Hello students, in this recording, we will learn homologous recombination in eukaryotes. This is the fourth recording of the chapter recombination of mandatory paper 2. So far, we have seen what is recombination, what is its significance, holiday model for homologous recombination, double strand break repair model for homologous recombination, the proteins involved in the DSB repair pathway in E. coli. And now we shift our focus to homologous recombination in eukaryotes. Homologous recombination in bacteria is required to repair double-stranded breaks in DNA, to restart collapsed replication forks, and to allow a cell's chromosomal DNA to recombine with DNA that enters via phage infection or conjugation. Homologous recombination is also required for DNA repair and the restarting of collapsed replication forks in eukaryotic cells. This is illustrated by the fact that cells with defects in the proteins that promote recombination are hypersensitive to DNA damaging agents, especially those that break DNA strands. Furthermore, animals carrying mutations that interfere with homologous recombination are predisposed to certain types of cancer. However, homologous recombination plays important additional roles in eukaryotic organisms. Most importantly, homologous recombination is critical for meiosis. We all know what is meiosis. During meiosis, homologous recombination is required for proper chromosome pairing and thus for maintaining the integrity of the genome. This recombination also reshuffles genes between the parental chromosomes, ensuring variation in the sets of the genes passed to the next generation or progeny. Homologous recombination is required for chromosome segregation during meiosis. Meiosis involves 2 rounds of nuclear division, also called as reduction division, resulting in a reduction of the DNA content from the normal content of diploid cells, which eukaryotes are, 2n, to the content present in gametes 1n. We all know gametes are haploid. If I talk about human gametes, sperm and ova, they are haploid and contain one set of chromosomes. Now we will see meiosis and how recombination takes place during meiosis. Before division, the cell has 2 copies of each chromosome, the homologs, one each that was inherited from its 2 parents. Rather than showing all the chromosome sets, they have shown only one chromosome in this picture for simplicity and for explanation purpose. So let's say this is chromosome 1, what you can see in blue color. And this is also chromosome 1, what you can see in red color. But this chromosome 1, which you can see in blue, let's say has been received from the male parent, and the chromosome 1, which you can see in red, let's say, has been received from the female parent. And if I show you this one parallel line is one chromosome. Here also the one parallel line is one chromosome. But why you see a X-shaped structure? Because these chromosomes are replicated to give rise to sister chromatids. So during the S phase, you know the cell cycle of eukaryotes, G1, S, G2 and M. During the S phase after gap 1, the synthesis phase, DNA replication takes place and these chromosomes are replicated to give a total DNA content of 4n. And the products of the replication, that is the sister chromatids, stay together. So if I said that the blue chromosome is chromosome number 1 received from the male parent, and the red chromosome is also chromosome number 1 received from the female parent, the blue chromosome has replicated, giving rise to a sister chromatid, and the chromosome which is shown in red is also replicated, giving rise to another sister chromatid. Then, in the preparation for the first nuclear division, these duplicated homologous chromosomes must pair and align at the center of the cell, which you can see in this step. How are they pairing and aligning? It is this pairing of homologs that requires homologous recombination. And these events are carefully timed. So what we have seen in the double strand break repair pathway, that these chromosomes, when they align and since they're homologous, double strand break takes place in one of them, the other remaining intact. Then the processing takes place to generate those single stranded tails, and then the strand invasion, holiday junctions, branch migration, and finally resolution. So this crossing over takes place, and recombination must be complete before the first nuclear division to allow the homologs to properly align and then separate. And these events are carefully timed. During this process, sister chromatids remain paired. You can see here the sister chromatids are paired. The red is pairing with red and the blue chromosome is pairing with blue. The sister chromatids are paired, but due to the recombination, you can see the blue chromosome had capital A and capital B alleles, whereas the red chromosome had small a and small b alleles. But due to this crossing over, what we have seen in this step, the small b allele has come on this chromosome with capital A, and the capital B allele has come on this chromosome with small a. And now one more division would take place, what we can see or we will see in the next step. Then in the second nuclear division, it is the sister chromatids that separate. You can see that the sister chromatids are being separated here, the blue getting separated from blue and the red chromosome separating from red. The products of this division are the 4 gametes, each with 1 copy of each chromosome, that is 1 N DNA content, and the gametes are haploid. And you can see here, this is same as parent, capital A, capital B. And this is also same as parent, small a, small b. But if you see this one, this is capital A and small b, and this is small a and capital B. So this is the result of the crossing over or the reassortment of genes have taken place. Without recombination, chromosomes often fail to align properly for the first meiotic division, and as a result, there's a high incidence of chromosome loss. This recombination is very important. It is essential, and without this, chromosomes fail to align properly for the first meiotic division, and as a result, there's high incidence of chromosome loss. This improper segregation of chromosomes called non-disjunction leads to a large number of gametes without the correct chromosome complement. Gametes with either too few or too many chromosomes, that is abnormal number, cannot develop properly once fertilized. Thus, a failure in homologous recombination is often reflected in poor fertility. The homologous recombination events that occur during meiosis are called meiotic recombination. Meiotic recombination also frequently gives rise to crossing over between genes on the 2 homologous parental chromosomes, which we have seen just now in the diagram. An important consequence is that the alleles present on the parental DNA molecules are reassorted for the next generation. Like you have seen how a small b allele has come with capital A and the capital B has gone with small a. Let's see the steps of this meiotic recombination. And in a double-strand break repair pathway, the first step is introduction of double-stranded breaks. And in eukaryotes, during meiosis, there is programmed generation of double-stranded DNA breaks that occurs. The developmental program needed for cells to successfully complete meiosis involves turning on the expression of many genes that are not needed during normal growth. So there are many genes that are not needed during the normal growth. But during the meiosis, or to successfully complete the meiosis, these genes should get turned on. One of these is SPO11. This gene encodes a protein SPO11 that introduces double-strand breaks in the chromosomal DNA to initiate meiotic recombination. So we know that the first step in a double-strand break repair pathway for homologous recombination is introduction of double-stranded break. And we've seen in the previous recording that there is no protein that has been found in E. coli, especially for this purpose. But in eukaryotes, there is a protein designated for this purpose. And this protein, SPO11, introduces double-stranded breaks in the chromosomal DNA during meiosis to initiate meiotic recombination. The SPO11 protein cuts the DNA at many chromosomal locations with little sequence selectivity, but at a very specific time during meiosis. So the SPO11 protein will cut the DNA, will introduce double-stranded breaks at many chromosomal locations during meiosis. Yes, sequence selectivity is there with little sequence selectivity, but more important than that is the time, at a very specific time during meiosis. And this SPO11 mediated DNA cleavage occurs right around the time when the replicated homologous chromosomes start to pair, which we have seen in the previous diagram. SPO11 cut sites are not randomly distributed along the DNA. The cut sites are located most commonly in chromosomal regions that are not tightly packed with nucleosomes, such as promoters controlling the gene transcription. Regions of DNA that experience a high frequency of DSBs also show a high frequency of recombination. Thus, the most commonly used SPO11 DNA cleavage sites, like Chi sites, are hotspots for recombination. Let's see the mechanism of introduction of double-stranded breaks by SPO11 in eukaryotes. A specific tyrosine side chain in the SPO11 protein attacks the phosphodiester backbone to cut the DNA and generate a covalent complex between the protein and the severed DNA strand. So in this picture, you can see there is a DNA here. This is the double-stranded DNA. And you can see this protein has come, which is SPO11. This is a eukaryotic protein. And it's a protein, it's made up of amino acids, obviously. And a specific tyrosine side chain of this SPO11 protein is going to attack the phosphodiester backbone of this DNA to cut the DNA to generate double-stranded breaks. And it generates a covalent complex between the protein and the DNA strands. In the first step, you can see this is the double-stranded DNA. And the SPO11 is approaching it. In the next step, you can see the SPO11 is bound to DNA. There are two molecules of SPO11. And this SPO11 ka tyrosine side chain is attacking this phosphodiester bond, is attacking the phosphodiester backbone, and it's introducing double-stranded breaks, which you can see here. But after the generation of these double-stranded breaks, the SPO11 doesn't leave the DNA. It has formed a covalent complex with the DNA and it's still there, which you can see. Two subunits of SPO11 cleave the DNA, which you can see here. These are the two subunits of SPO11. They cleave the DNA two nucleotides apart on the two DNA strands to make a staggered double-strand break. Yes, it has to be a double-stranded break because it's DSB repair pathway. And there are two subunits of SPO11 involved. And they cleave the DNA two nucleotides apart. You can see in the upper strand and in the lower strand, the cleavage is not, or the cut is not at the same position. So it cleaves the DNA strands two nucleotides apart to make a staggered double-stranded break. In the third step, they're actually shown the tyrosine of this SPO11 and how this tyrosine side chain of the SPO11 protein is attacking the phosphodiester backbone of the DNA, making a cut, how two molecules of SPO11 are involved, and they're making the cuts two nucleotides apart, giving rise to a staggered double-stranded break. This protein has not left the DNA, it's still making a covalent complex. The reason is, we'll shortly see, SPO11 shares this DNA cleavage mechanism with the DNA topoisomerases involved in supercoiling and site-specific recombinases. In fact, SPO11 appears to be the distant cousin of these enzymes. The 5' ends of the DNA at the site of the SPO11 cleavage are covalently bound to the enzyme, which we have seen that the SPO11 doesn't leave after it has made the cuts. It's covalently bound, to the DNA, or the DNA is covalently bound to the enzyme. It is these SPO11 linked 5' DNA ends that are the initial sites of DNA processing to create ssDNA tails required for assembly of REC-A-like proteins and initiation of DNA strand invasion, which is the next step. Second, the energy of the cleaved DNA phosphodiester bond is stored in the bound protein DNA linkage. So when this SPO11 makes cuts in the DNA, it remains covalently attached to the DNA because the energy which is released when this DNA is cleaved, the energy is stored in the bound protein DNA linkage, and the DNA strands can be resealed using the same energy, which will be a simple reversal of this cleavage reaction. And this resealing will occur, that is, the cuts which are made will be resealed again when the cells will receive a signal to stop proceeding with meiosis. After SPO11, we move on to the next protein. So if you see the DSB repair model, the first step is alignment and introduction of double-stranded breaks. So in prokaryotes, no protein has been found that introduces double-stranded breaks. It may be due to DNA damage in this. Here in eukaryotes, during meiosis, SPO11 introduces double-stranded breaks in a very accurate and a programmed manner. After the generation of double-stranded break, the next is processing at these broken ends to generate single-stranded DNA regions or single-stranded DNA tails ending with 3' end. In eukaryotes, the protein which does this processing is MRX. The MRX processes the cleaved DNA ends for assembly of REC-A-like strand exchange proteins. The DNA at the site of SPO11, the DNA at the site of the SPO11 catalyzed double-stranded break is processed to generate single-stranded regions needed for assembly of REC-A-like strand exchange proteins. As was observed in the REC-BCD pathway of E. coli, this processing generates long segments of single-stranded DNA that terminate in 3' ends. During the meiotic recombination, the MRX enzyme complex is responsible for this DNA processing event. How we have seen in E. coli pathway that the enzyme that does this processing to generate the single-stranded DNA tails is REC-BCD. Here in eukaryotes during meiosis, the enzyme is MRX. Though it's not analogous to REC-BCD, MRX is also a multi-subunit enzyme and it's a DNA nuclease. MRX is composed of 3 protein subunits called MRE11, RAD50, and XRS2. The first letters of these 3 subunits, MRE11 ka M, R of RAD50, and X of XRS2 gives the protein its name, MRX. It's a multi-subunit containing 3 subunits, MRE11, RAD50, and XRS2. Processing of the DNA at the break site occurs exclusively on the DNA strand that terminates with a 5' end. That is, the strands covalently attached to the SPO11. So where the SPO11 is attached, that is the strand which will be processed at the break site. Exclusively that strand will be processed that terminates with 5' end, the one which is covalently attached to SPO11. The strands terminating with 3' ends are not degraded. So one of the strand is degraded, one of the strand is not degraded. Same way what double-strand break repair predicts. This processing reaction is therefore called 5' to 3' resection, and the MRX dependent 5' to 3' resection generates the long ssDNA tails with 3' ends, and they are often 1 kb, that is 1000 base pairs or longer. The MRX complex is also thought to remove the DNA-linked SPO11. The SPO11 will be removed from the DNA which was bound to DNA so far by the action of MRX. After SPO11 and MRX, we come to the next protein of eukaryotes. So again, try to recollect the DSB repair pathway. First step is alignment. Next is introduction of double-stranded breaks, which is done in eukaryotes by SPO11. Next is processing of the ends to generate 3' single-stranded DNA tails, which is done by MRX in eukaryotes. Now the next is strand invasion. The strand invasion in bacteria E. coli is done by Rec-A, which is loaded by the Rec-BCD. Here, the strand invasion is done by DMC1. It's a Rec-A-like protein that specifically functions in meiotic recombination. Eukaryotes encode two well-characterized homologs of the bacterial Rec-A protein, which is involved in strand invasion. These two proteins are RAD51 and DMC1. Although the title says DMC1, RAD51 is also a Rec-A-like protein, which is involved in recombination. We'll see shortly why DMC1 is emphasized here. Both proteins functions in meiotic recombination, RAD51 and DMC1, whereas RAD51 is widely expressed in cells dividing mitotically and meiotically. DMC1 is expressed only as cells enter meiosis. So RAD51 is expressed in cells dividing mitotically and meiotically. DMC1 is expressed only as cells or when the cells enter meiosis. DMC1 dependent recombination is preferentially between the non-sister homologous chromatids rather than between the sisters. So this DMC1 dependent meiotic recombination is between the non-sister homologous chromatids rather than between the sisters. We'll see this with the help of a diagram. So here you have two DNA molecules, one shown in green, one shown in purple for the explanation purpose, and these are the non-sister homologs. Now you know what are non-sister chromosomes. Alignment has happened. The first step after alignment would be introduction of double-stranded breaks. That would be done by SPO11, which you can see in the picture. SPO11 has come, the tyrosine side chain has attacked the phosphodiester backbone, generated double-stranded breaks, two nucleotide apart, so staggered cut, and the SPO11 remains attached. Then MRX comes into picture, and MRX, which is a nuclease multi-subunit combination of 3 proteins, comes and start processing the ends. The MRX has processed, you can see in the next step, to give rise to this single-stranded DNA tails ending with 3' ends. Not only that, due to the action of MRX, the SPO11 has, which is covalently bound to DNA, has left the picture. This is called as 5' to 3' resection by MRX, which is this generation of single-stranded DNA tails. So far, SPO11 has introduced the double-stranded breaks, and MRX has done processing to generate 3' single-stranded DNA tails. Next, you can see RAD51 and DMC1 has come into picture, and the MRX will leave. And the RAD51 and DMC1 have coated these single-stranded DNA tails. How this RAD51 and DMC1 are loaded onto these tails, we will see that also shortly, like how Rec-A is loaded by Rec-BCD in E. coli. After these tails are loaded with DMC1 and RAD51, they will do the classic strand exchange, because the strand exchange protein filaments are formed. And you can see how this strand, which is shown in green color, is separating the unbroken DNA molecule, which are the two purple strands, and it's pairing with its complementary strand in the unbroken DNA, and the other purple strand will pair with this second green strand. So this is strand invasion, and which is mediated by DMC1 and RAD51. So you can see here these are the chromatids they have shown. So there are 4 chromosomes in total if you see here. Let's say this blue color is chromosome number 1. And this red is also chromosome number 1. Blue is from one of the parents, red is from another parent. They have replicated to give rise to one more copy. There are 4 chromosomes. These are the 2 which have replicated. The 2 blue chromosomes are the sister chromatids. Similarly, the 2 red chromosomes are sister chromatids. You can see the interhomolog recombination by DMC1 is preferentially between the non-sister homologous chromatids. The majority of the DNA sequence among these 4 copies will be identical. But as we said, DMC1 recombination is between the non-sister homologous chromatids rather than between the sisters. Although the basis of this selectivity is unknown, there's a clear biological reason that meiotic recombination promotes interhomolog connections to assist the alignment of the chromosomes for division. Many proteins function together to promote meiotic recombination. So far, we have seen SPO11, MRX, and RAD51 and DMC1. But there are other proteins also that function together to promote meiotic recombination. Proteins involved in the critical stages of double-strand break formation, generation of 3' single-stranded DNA tails, and strand exchange during meiotic recombination have been identified and characterized. Genetic experiments indicate that many additional proteins also participate in this process. Furthermore, many proteins appear to interact with the known recombination enzymes, and it seems likely that these proteins function in the context of a large multi-component complex. These large protein DNA complexes, known as recombination factories, can be visualized in cells. RAD52, if I take you back to the MRX components, MRX is composed of MRE11, RAD50, and XRS2. So RAD50 is one of the components of MRX. Then RAD51 is involved in strand invasion. But there's one RAD52 as well. And this RAD52 is another essential recombination protein that interacts with RAD51. RAD52 functions to promote the assembly of RAD51 DNA filaments, the active form of RAD51. And this RAD52 does this by antagonizing the action of RPA, which is a major single-stranded DNA binding protein present in eukaryotes. So here also, it is ensured that the SSB should not bind to the single-stranded tails first. And in fact, the protein which is involved in strand invasion, RAD51 and DMC1 here, RAD51 should bind to the single-stranded DNA tails. So the RAD52 is doing two things. It is not allowing the single-stranded DNA binding protein, which is RPA, to bind to the single-stranded DNA tails, plus it's directly promoting the assembly of RAD51 onto the single-stranded DNA tails. So in this respect, RAD52 shares an activity with the REC-BCD protein. REC-BCD, how loads REC-A onto the ssDNA and not allow SSBs to bind. That would otherwise have been. Yeah, so REC-BCD helps load REC-A onto the DNA that would otherwise have been bound by SSBs. So, but if we compare eukaryotes with E. coli, the broken ends are processed to generate single-stranded DNA tails by REC-BCD, and the REC-A is also loaded by REC-BCD. So REC-BCD is doing both the things same. But if you see eukaryotes, the broken ends are processed to generate single-stranded DNA tails by MRX, whereas the strand invasion or the RAD51 protein is loaded by RAD52. If you see or compare eukaryotes with bacteria, we expect that eukaryotic cells encode proteins that promote the branch migration and holiday junction resolution steps of recombination. In fact, enzymes capable of promoting these reactions are being identified. And for example, the MUS81 protein, which is highly conserved in eukaryotes, is required for meiosis and will function as a holiday junction resolvase. So we have seen in this video homologous recombination in eukaryotes, how homologous recombination takes place during meiosis and why is it important. And what is the outcome of that? Then we have seen SPO11, MRX, DMC1, RAD51, RAD52, and MUS81. Now, to conclude this recording or to summarize, we will again see the DSB repair model. We'll see which of the each of the proteins of the eukaryotes acting at these steps. So if we see the first step, alignment, then there's introduction of double-stranded break. No protein has been found in E. coli for this. It's due to damage or, stalled replication fork. But in eukaryotes, there is a special or a specific protein which introduces this double-stranded breaks by making staggered cuts, that is SPO11. Then the next is processing to generate this single-stranded DNA tails with 3' ends. And this is done by MRX, which is a multi-subunit nuclease having 3 proteins. Then the strand invasion protein is loaded onto these single-stranded DNA tails, and the protein that loads the strand invasion protein onto these ssDNA tails is RAD52. Then the proteins which do this strand invasion, which you can see on this slide, is DMC1 and RAD51. Branch migration, many proteins have been identified, and we assume that there are proteins involved in branch migration as well in eukaryotes. And the last protein which will make this resolution or resolve is MUS81. So this was all about homologous recombination in eukaryotes or meiotic recombination and all the proteins involved in the same.